Hello, my student. I'm your biology teacher, Cikgu Govin. Topic today is leaf structure and function. The subtopic is 2.4, main organ for photosynthesis. Here are the learning standards for subtopic 2.3, main organ for photosynthesis. There are 9 learning standards, but in this video I cover learning standard number 1, number 2, and number 3. 4, 5, 6 I cover in next video for series B, and 7, 8, 9 I cover in video series C. So today just learning standard number 1, 2, 3. Okay. So number 1 is justify the necessity of photosynthesis in plant. Number 2 relates the adaptation of internal structure of a leaf to photosynthesis. Number 3 identifies structure of the chloroplast, which is granum, thylakoid, and stroma. The necessity of photosynthesis in plants. Plants are autotrophic organisms. This means the plant can produce their own foods through a process of photosynthesis. So this is the product of photosynthesis, glucose. The product of photosynthesis, which is glucose, is used by other organisms to generate energy through oxidation of foods. So plants not only produce glucose for their own, but also for other organisms. Energy is needed to carry out living processes such as growth and reproduction. Through oxidation of foods or through oxidation of glucose, energy is generated. So in cellular respirations, this energy is needed for carry out the living processes. Number 4, plant release oxygen, resulting in a symbiotic relationship between plant, animal, and humans. What is meant by symbiotic relationship between plant, animal, and human? You see the chemical equation here. The product of photosynthesis is oxygen. So what is the byproduct process of cellular respiration in human and animals? The answer is carbon dioxide. So carbon dioxide is needed in process of photosynthesis in plant. So that's meant by symbiotic relationship between plant, animal, and humans. Both are dependent to each other in this relationship. Brief history discovery of photosynthesis. This is experiment conducted by Jean Baptista van Helmont. So he scientist from Belgium. This is the experiments conducted by him. First, he wet the seedlings, and then second step, he wets the dry soils. So he watered the plants for almost 5 years. After 5 years, he found out that the plant weight is 77 kilograms, but the dry soil still remain the same, 90 kilograms. So the conclusion of his experiment is the plant had grown by only being watered and not from the water of the soil. So this is the contribution of Van Helmont in discovery of photosynthesis. Okay, another one scientist that play important role in discovery of photosynthesis, Joseph Presley. Okay, from this diagram you can see that there are two inverted glass jar. This one you can see that red was placed in an inverted glass jar, the red died. But the second inverted glass jar, there is a plant in here, the plant in the inverted glass jar, the red leaf. So from these experiments, Presley concluded that plant release oxygen. So this is example of symbiotic relationship between plant and animals. The adaptation of the internal structure of a leaf to photosynthesis. Photosynthesis needs light energy, carbon dioxide, and water. So photosynthesis needs chlorophyll to absorb light energy from the sun, and also carbon dioxide from atmosphere and water from the soil. So the byproduct of this process are oxygens. Besides the leaf being the main photosynthesis organ, young stem and other green part of the plant are also able to carry out photosynthesis. Let us study the cross-section of a leaf. Number one is upper epidermis. Okay, this part is upper epidermis. The second layer is palisade mesophyll. This one arranged vertically closely packed, okay, to allow maximum absorption of light. So here abundance of chloroplast here. And number 2B is spongy mesophyll, loosely arranged, okay, so that the gaseous actions can occur efficiently. Number 3 is lower epidermis, okay, this part is lower epidermis. Here you can see the opening here. This is stroma to allow gaseous action and water vapor to diffuse through it. And number 4 is vascular bundle. The pink color is xylem, okay. The blue color is phloems. Now we study the adaptation of the internal structure of a leaf to photosynthesis. Upper epidermis and lower epidermis, number 1 and number 3. Okay. Transparent waxy cuticles on the upper and lower epidermis allow sunlight to penetrate to the palisade mesophyll and also to the spongy mesophyll. Don't forget that spongy mesophyll also contain chloroplast, but more chloroplast in the palisade mesophyll. Stomata are present in the lower epidermis. In the presence of light, stomata open and allow gaseous exchange to occur. And don't forget also, there is chloroplast in the gut cell. Gut cell control the opening and closing of stomata. Palisade mesophyll. Palisade mesophyll packed with chloroplasts. This allows maximum absorption of light energy. Chloroplasts contain chlorophyll. Actually, chlorophyll is the pigments that play an important role in absorption of light energy for photosynthesis. Spongy mesophyll. Spongy mesophyll contain lesser amount of chloroplast compared to the palisade mesophyll. Here is spongy mesophyll, loosely arranged, so contain A spaces. So this allows efficient gaseous actions during photosynthesis. Vascular bundle, number 4 here, consists of xylem and phloems. Xylem, the main function is to transport water and mineral salt absorbed from the roots to the leaf. And phloem, it functions to transport sucrose produced by photosynthesis from the leaf to all parts of the plants. Okay, this is xylem and this one is phloems. Okay, this is chloroplast structure. A chloroplast consists of thylakoid, grana, stroma, and lamellae. So this is thylakoids, one disk of thylakoids. A group of thylakoid, we call it granums. Okay, this is stroma, the colorless fluid that's surrounding the granums. Okay, this is lamellae that connect one thylakoid to another thylakoid. And this chloroplast structure is coated by 2 layers of membranes, outer membranes and inner membranes. Okay, we see again chloroplast structure. Can you label it? Okay, we do it together, okay? So this one is outer membranes, inner membranes, lamellae, stroma, thallicoid, and granum. Thallicoid. Thallicoid, the structure looks like this shape and it contains chlorophyll. In thallicoid membranes, there are photosynthetic pigments that traps sunlight energy. Light-dependent reaction, or known as light reactions, occur in thallicoid. Granums, a dish-shaped stack of thallicoid. This arrangement is to increase the surface area so that the process of photosynthesis can occur rapidly and efficiently in the chloroplast. Stroma. Stroma is the colorless fluid surrounding granum in the chloroplast. This is the site for light-independent reaction, or also known as dark reactions, to take place, which produce glucose, the product of photosynthesis. Why do leaves change color in autumns? In four-season country, the green color of leaf can change to yellow, orange, red, or browns. This is because of the changes in light intensity, temperature, and weather. The chlorophyll breakdowns, the green color disappear and change to other pigments. How many types of photosynthetic pigments in leaf are involved to produce the green color of a plant? So we need to use chromatography methods to separate type of photosynthetic pigments in the leaf. So chromatography is a method of separating components of a mixture based on the differences in solubility of components in certain solvents. Activity 2.3, activity of chromatography. The aim of this activity is to carry out an investigation to separate photosynthetic pigments in a leaf using paper chromatography. Here are the material: Colliers plant leaf, pandan leaf, red spinach leaf, fine sands, 80% acetones solvents, 1 part of acetones and 9 part of veteran ether, skewer, filter paper, and pencil. The apparatus is glass container, ruler, mortar, and pestle. Procedure: Number 1. Ponds 2 leaf of Colliers plants, pandan leaf, red spinach leaf with 80% acetones and a little bit of fine sand using a mortar and pestle until a thick leaf extract is obtained. Number 2. Prepare a filter paper of size 3 cm x 15 cm. Number 3. Draw a line of 1.5 cm from the end of the filter paper horizontally using a pencil. Number 4. Transfer the leaf extract in the middle of the pencil line and let it dry. Number 5. Repeat step 4 10 times to get a thick extract on the filter paper. Number 6. Dip the end of filter paper in a glass container containing solvent at the height of 1 cm. Number 7. Let the filter paper dry for a while and absorb the changes. Number 8. Mark the point of each pigment that can be seen on the chematograms. So this is the setting of the paper of chromatography: skewer, glass container, filter paper, pigment of leaf extract, we put it here. This is pencil lines for indicator where we start the measurements, and this is the solvents. This is the Result of activity 2.3. From the diagram, you can see this is the loading lines when we put the leaf extracts. Okay, the solvent carry the dissolved pigments as it move up at the paper, and then the pigments are carried along at a different rate because they are not equally soluble in the solvents. So this is the result. The pigments, carotene, xanthophyll, chlorophyll A, and chlorophyll B. There are 4 pigments in extraction of spinach leaf. So discussion question, what is the function of solvent in this activity? To dissolve chlorophyll. Number 2, why is fine sand used to prepare the leaf extracts? To break down the leaf cell to make chlorophyll extraction easier. Number 3, how are the photosynthetic pigments in the leaf separated using the chromatography methods based on the value of R, which is the ratio of the distance traveled by the pigments to the distance traveled by the solvent along the chromatograms. So this is actually the formula they use to measure the type of the pigments that found in the spinach leaf. That's all for today. See you again in next lessons, but we still continue to these subtopics, learning standard 4, 5, and 6 in series video B. So that's all. Thank you and bye-bye.